Okay, I think we can start. I hope you've all had a good lunch. And it's my honor to have been asked to chair the last and final session of this meeting that's actually been a continuity for a number of days, and I'm very sorry that I wasn't able to attend at the Dead Sea. So my name is Karen Abraham. I'm the Vice Dean at the Sackler Faculty of Medicine, just a few meters down the street. And this is an issue that we all deal with. I'm sometimes a little surprised we're not that many people, and yet I would say that everyone at the university, the 30,000 students, the faculty, everyone else that works here is having to deal with this concept. So I'm hoping that from the past days and from the continuity of the day today, that there'll be a lot of this brainstorming that I know that Mira really wanted you to have, us to have, so that we can come up with strategies and ideas so that the 30,000 beyond this room can really benefit, and more than that. So I wish you a very successful session.
So before you
yes,
present,
please.
You need to do something in the computer.
Okay. Okay. So please, you can have the computer, and then I'll introduce the first speaker. Okay. We'll start. Okay. So I will. It's my tremendous honor, really, to introduce Professor Ilana Gozes, who's not only a very close colleague, but also a very close friend. And I will tell you that she's one of the major reasons that I'm here at Tel Aviv University. So we won't talk about how long ago that was, just a few years ago. But Ilana was really instrumental, so that's a personal thank you. But Ilana has done tremendous work over the past years on a molecule that she discovered years ago and has really taken it from a very small one molecule to an enormous story that has implications for not only children's health, but a lot beyond, as you'll see when she talks about the implications in the aging population. So Ilana, please.
So first of all, thank you very, very much for this wonderful introduction. And if I can open my presentation, I cannot talk without slides. Kind of. So I prepared a presentation that is for scientists, but I was also thinking about the lay audience, so I hope that I won't be too boring to the scientists or too boring to the lay audience. And I'm going to talk to you today about a molecule that we discovered that we called ADNP in short, or in long, activity-dependent neuroprotective protein. So as a child, I was interested in the brain and in genetics. So if we think about the brain, we have 100 billion nerve cells. And then we actually have more support cells. So the nerve cells talk to the support cells. So that was one of the questions that I've asked quite a number of years ago. And then we have 100 trillion synapses. So synapses are the point of connection. So and we heard this actually this morning also. So each nerve cell makes many, many connections. So if something goes wrong, then the system, it's like as I draw it, it's like the whole train system goes berserk. So can we fix it? And now from the generalization, I'm going to talk to you about the details. So we kind of look to see what protects, how do the brain cells talk to the support cells, and is there any support, is there any protection from the support cells to the brain cells? And that's how we discovered ADNP, or in short, or in long, activity-dependent neuroprotective protein, because it protects nerve cells against electrical blockade, which happens in neurodegenerative diseases, for example. And we also derived a very small component, a very small fragment of ADNP that we now know enhances ADNP activity. And why is ADNP so important? So we discovered it as nerve cell protector against electrical blockade. And then we knocked it out in animals, and my student, who is now the vice president of research and development in Tel Aviv University, Professor Albert Pinchasov, came to me and said, I'm not getting any animals, they die. So I said, let's look and see why they die, and they don't form a brain. So if there is no ADNP, the neural tube doesn't close. So it's a no-brainer. It is required for the brain formation. And now, like after many years, we know that ADNP mutations, de novo mutations that occur out of the blue during pregnancy, cause autism-like syndrome in children, and it's now called the ADNP syndrome. And these are a few children that suffer from the ADNP syndrome. So this is, you know, when I was a child, we didn't know why children are autistic. We just called them the strange kids. We didn't know. And now we know that many of these autistic children actually, or within the spectrum, I should say, actually have a single mutation in a single gene, and ADNP is one of the leaders. And then there is this phenotype of severe speech delay, intellectual disability, motor development delay, sensory processing disorder, insensitivity to pain, and sleep disorders. And so this is for the more aficionados. This is the ADNP gene, and here you see some of the mutations. But there are actually quite a number of more mutations, and every day there is another mutation that is being found out. So originally, like 3 years ago, there were 2 children, and now there are 150 children, and the number is expected to grow quite dramatically, and it is specific, the ADNP gene is specific to higher organisms. To vertebrates, to chordates. So because it is important for the formation and the shaping of the brain. And here you see some more mutations in the ADNP gene, and it regulates 400 genes during development, which are key to the development of the brain. And it also regulates genes that are associated with bone formation, because these genes are actually shared by the nervous system and by the bone system. So there are shared genes. And one of the diagnostic procedures that we have discovered together with one of the mothers who has twins, and you see here the twins. So this is the ADNP child, and when he was 1 year old, he had a mouth full of teeth. His brother had a mouth full, empty of teeth. And also, when they shed the teeth, it was earlier in the ADNP child, and the teeth are with less enamel. So the mother described this as like shark teeth. So actually, just by one way of diagnosing these children is to see when the teeth come out. And with these children, the teeth come out very early and sometimes several together. So this is one way of early diagnostic, which is very, very cheap. The other way is, of course, to do a complete sequencing of the coding sequences of the entire genome of the mother, the father, and the child, and this is called TRIO, and then it's like finding a needle in a haystack, find the mutation. So this is how these children are diagnosed. It's called whole exome sequencing. And here you see, so this is done by others, and now this is big data, looking at many, many, many autistic children, and and each one has a different gene mutation, and then looking what are the prevalent genes. So ADNP is a star here. It's one of the most prevalent genes that is mutated, and then there is autism-like syndrome. And here you see again 2 children, and you see this child again. And so we calculated the prevalence to be something in the kind of developed world, in the USA, EU, and Japan, to be something like 6,200, but maybe it's even a little bit more. We have 1 child in Israel, and you see here the distribution. And the distribution is like that because these are the countries that actually give this whole exome sequencing diagnosis, and then the children can be diagnosed. And so there is still all this world that needs to do that as well. And so now it's a little bit more science. So apparently these genes that are associated with mutations in autism and causing autism can be actually found grouped together, and some of them physically interact. And as you see here, for example, the most common autistic-like syndrome is Fragile X syndrome. This is actually inherited, and the Fragile X gene interacts with kind of indirectly with ADNP, also the gene that causes the Rett syndrome, which is also a de novo mutation. The MECP2 also interacts with the same complex that ADNP interacts with, and also another gene that is called disrupted in schizophrenia, so you can think about what disease is associated with this, this is also inherited, and this also interacts with the same kind of proteins that are all together regulate the expression of other genes, and they are called chromatin remodeling complex. So they are within this complex. And here you see again some children with the ADNP syndrome, and here I just wanted to emphasize, first of all, there is 20% of the children right now have one single mutation which truncates the protein in the middle. And one thing also to say is that all the children are all heterozygous, meaning they have one normal allele and one truncated protein. So it's a single point mutation, and then all of a sudden the protein is truncated, it's shorter, and then we have this sequelae. And you see here that they start walking when they are like 3.5. And some of the parents are actually told that the children won't walk at all. So they start with the aids, and then eventually they walk. And there are, when you look at the MRI, there are deficits in the brain, especially in the area of the hippocampus, which is important for memory formation and learning. And so ADNP, we are studying it now for about 20 years, and we know what protein it interacts with. So actually, when it was found to be mutated in autism, the researchers knew quite a lot about it because of all our work, but we were kind of working on it solo. Now I have some collaborators and competitors. And it interacts, as I said, with the chromatin, so with the DNA and gene expression. It regulates, so now I spoke about autism, but one of the major genes that it regulates is called apolipoprotein E. And apolipoprotein E, we have 3 different forms of it, 2, 3, and 4. People who have the apolipoprotein E4 are more prone to get Alzheimer's disease. Right now, we don't want to know exactly because we don't know exactly how to stop it, but that's no. So ADNP regulates apolipoprotein E, and it also is associated with a system in the cell that is called the microtubule system, and without the microtubule system, which is the skeleton of the cell, the nerve cell will actually collapse because the nerve cells has very long processes, and there it makes the connections, and without the microtubules, it won't work. And then it also, and this system, the microtubules, which is the skeleton, the scaffolding, and also like the transportation system of the nerve cells, they also interact with another system in the cell, which is the cleaning system, or it's called also autophagy, so eating themselves, the cell self, but it's eating all these proteins that precipitates and cause the degeneration, for example. And actually, the Nobel Prize was awarded this year for the discovery of autophagy. So just to quickly, so we found that ADNP regulates like 400 genes during development, thousands of genes during postnatal development. This is all in the mouse. And one of the genes that it regulates is the subunit protein of the microtubules, of this skeleton and transport system of the nerve cells, as you can see here. And it does so in a sex-dependent manner. And this is also interesting because, as you know, women, for some reason, are more prone to Alzheimer's disease, and boys are more prone to autism. And it is kind of in the middle. And so when we looked at ADNP and we were thinking in terms of medical progress, it's a very, very large protein. And that actually, it is common to genes that are mutated in autism. They are large. So, autism de novo, because then there is a possibility of getting all of a sudden a mistake. So in terms of developing ADNP itself as a drug, it's going to be very expensive and very difficult to protect the brain. So we started working on the protein and made small fragments. And one fragment that we made, which we called NAP, and now it's called also CP201, it has some additional names. So this peptide, or this small protein, small fragment, actually mimicked some of the ADNP activity. By that I mean if in cells, so these are neuronal-like cells, and what is characteristic of a neuron is the long processes. You see the red long processes. If we knock down ADNP genetically, we lose this. So ADNP is not only required for the entire brain, it's also required for the processes of the nerve cell to make connections. We knock it out, it disappears. And similarly, if we take nerve cells and expose them to oxygen glucose deprivation, like in stroke, for example, or like in aging, when we have too little oxygen, too little glucose, we lose this microtubular system in red, which makes these processes, these axons. And then when we add the small fragment from ADNP, we actually build it again. And it's, it's a visual experience. And so now we know precisely how this is done. So we know NAP, our CP201, has this sequence. So this is actually now amino acid sequence. This is the building blocks of a protein. And this sequence interacts with the growing ends of the microtubules. So when I say that this is the skeleton, it's a dynamic skeleton. So it's not like in a building that you have a tube and it stands there, but it's a dynamic tube that it is building and dissipating from the other side. And what you see here in green is the building of it. And what ADNP and the small fragment of ADNP are doing, they are actually attaching here and building the microtubules and then improving nerve cell health, actually improving growth and connection and synaptic connection. And here is the proof. We can actually take a nerve cell in culture and add to it NAP. This is our small fragment from ADNP. And you see all these green dots are actually points of connections. It is the dendritic spines that make the connection between nerve cells. So that's when you think about the children that have intellectual disabilities, then you understand if they have, the ADNP is not functioning, they have less synaptic connections, they can make less synaptic connection and hence the problems. And also when they grow slower, and they don't walk because they don't make the synaptic connection fast enough because they don't have enough ADNP. And here is kind of the proof. And so what happens in Alzheimer's disease, this is the other side of the spectrum completely. So in Alzheimer's disease, one of the major things that happens is the microtubules break down. And actually, it happens in regular aging, so I don't want to age. so what you see here the microtubules, and it's microtubules, and they have a protein that actually enhances the polymerization, enhances the formation of microtubules, and it's called tau, like Tel Aviv University, or tubulin associated unit. And this tau falls off the microtubules and forms what Dr. Alzheimer saw, and didn't know what it's made of. And these are the neurofibrillary tangles. And these are associated with cognitive decline. And this is what is found in the brains of Alzheimer's patients. So this is not my work. This is the work of George Perry, who is the editor-in-chief of the Journal of Alzheimer's Disease. And what you see here is that when we age, normally, our number of microtubules is decreasing. So these are not such good news. And tau, on the other hand, actually precipitates. So you see it here. So the microtubules, but when we normally age, the microtubules are shorter, but when we have Alzheimer's disease, they're even shorter. So Alzheimer's disease is like an accelerated aging, and then we get cell death. And actually, when we speak, we say that if everybody will live to 200, everybody will have Alzheimer's disease eventually, maybe. So even though when I gave a talk and I told this to somebody in my mother's assisted living home, one of the guys said, I am 92 and I don't have Alzheimer's. Am I over it? I said, yes. So just to be a little more into the mechanism, so we have, so now I'm going more into the molecular mechanism, so this is what I do for a living. And so we have the microtubule end binding protein, we have the microtubules, and we have the tau. So what was discovered is this microtubule end binding protein actually bind to tau. And we know that our small peptide, small fragment from ADNP, binds to the microtubule end binding protein. So what we found is that actually there is a crosstalk here. So I'm not reading you all the questions, I'll just show you the answers. So what you see here, and again, it's a visual experience, you see here the movement of the microtubules, very slow. You add NAP, very fast. So we actually change the movement of the microtubules. We change the dynamicity of the nerve cell. We make it more alert. And since this is a small fragment from ADNP that enhances ADNP binding to these organelles, we need ADNP there to make sure that everything is moving correctly. And if everything is not moving correctly, it's a problem. And the other thing is that we know that ADNP protect cells. And in order to protect the cells, it needs tau. So here we took cells that don't have tau, we put tau in them, and then we measured how do they survive in the presence of zinc. So zinc is actually a bit of a killer. If you have it too much, it is also found in the plaques. And the only cell that remained alive, as you see here, the green things are the cells, is the cell that had both tau and NAP. So you need both for survival. And the other thing that we did a lot of biochemistry, and what we found, and here it is kind of a visual experiment, is that when we have both NAP, or CP201, and microtubule N-binding protein and tau, actually tau is driven to the microtubules. So it will enhance microtubule stability rather than precipitate and makes these neurofibrillary tangles that are associated with cognitive decline. So we know the mechanism. And we are pleased about it. I hope that you understood me. And then we can also measure axonal transport. So we can measure transport of material in, it's again, the animal brain, and see how it moves. And this is very important for the cell to be alive, to communicate with other cells. And when I say cell, I mean the nerve cell. And we do it by giving the animal, this is the mouse, to inhale manganese, and the manganese actually moves, and it is white. And you see it here, it moves in the nose, it moves in the olfactory bulb, and it is white, and you see the movement. So what we discovered is that if there is a paucity in ADNP, so we have animals that have half the content of ADNP. Without ADNP, they don't have a brain. If they have half the content, they're a little bit like the autistic children. And then the transport of materials in the axons is much slower. It's actually almost, here it is almost non-existent. You see, it's very, very slow. This is the normal, this is the haploinsufficient, the half the content. When we give these animals a very small fragment of NAP, so it's a smaller fragment of ADNP, and we call it SKIP, we can actually bring it almost to normal. And what we also found is that there is a difference between males and female, and we are fast. But everything else is the same. So the brains maybe look the same of males and females, but there are subtle differences, both in gene expression and also in transport of materials between the male and the female mouse brains, and probably also in the human situation. And then when we look at these animals that have half the content of ADNP, and we put them in a learning paradigm. So the learning paradigm is they have to remember where the dry platform is in a water maze. So this is called the Morris water maze. So you see this is a brilliant mouse. He reaches the platform in 1 second. And this mouse takes a very long circuitous route in red to find the hidden platform. And what you see here is that animals that have half the content of ADNP, they actually, after 5 days of training, they don't find the platform. Because we measure just the time it takes them to reach the platform, they just don't find the platform. Normal animals, after 5 days, they know where the platform is. If we treat these animals intranasally for 2 weeks with NAP, the small fragment of ADNP, so it's like a replacement therapy, and they learn. And this is associated with an increase in tau pathology, which is measured in tau, actually hyperphosphorylation. So addition of phosphorus groups actually makes the tau more like fall off the microtubules and precipitate into tangles, and this we see here. So I'll go faster. And the other thing that we saw is that actually so I mentioned that ADNP regulates apolipoprotein E. So we asked, is this regulated the same in males and females? And we see no. It is more regulated in the females. And as I said, apolipoprotein E is a risk factor for Alzheimer's disease. So here we find that the regulation by ADNP in males and females differs. It's more regulated in the females. And we also saw in post-mortem material that there is more ADNP in the male, and this is like middle-aged, male brain, male hippocampus, which is the area where we learn and remember, than in the female. And the other thing we saw is this gene that is called eukaryotic initiation of factor 4E, which has been linked to autism is also regulated differentially by ADNP and more regulated in the males, in the young males, than in the females. Now, this is mice, but this is interesting. And now I'm going more into the Alzheimer's story. So somebody else, Young, did a study, Young et al. Did a study and looked at all the serum protein in Alzheimer's disease patients early on versus normal controls. And the only protein that he found that is going down in the serum is ADNP. So now this, we don't think with our serum, but this is interesting. We don't know how much of this ADNP can go into the brain or not, but it is still interesting. And ADNP not only is the regulatory in the brain, but it may also regulate our inflammatory response, because if there is less ADNP, there would be more inflammation. And this is also some studies that were done by others and also by me. And we looked at normal individuals. So there is a cohort in Harvard Medical School where they collected people. So these are people from Boston area, so they're all very educated, minimum 13 years of education. But these people have parents who have, one or two, who have Alzheimer's disease. So they are at risk. We don't know exactly why, because it's not genetic, but they're in a bit of a greater risk. And when we measured their ADNP levels in the serum versus, and they measured their IQ, we saw a correlation. And if it's age-adjusted, we saw actually a significant correlation. So there is some kind of a correlation between the expression of ADNP in the blood and IQ, and also afterwards maybe the development of Alzheimer's disease. So this is something that we would like to follow on. This is a recent study. And another study that was performed is with this. So a number of years ago, I started the company. I called it Alon Therapeutics. And this small compound from ADNP, which we now call also clinically CP201, was tested in a clinical trial in the United States in people that have amnestic mild cognitive impairment. So these people are actually normal. They still function, but they suffer from cognitive decline for memory problems. And they were enlisted into the study. So there were 144 people. And they were divided into 3 groups. So one group got a placebo. One group got low dose of our compound, CP201, either just once a day. And this is for 12 weeks. And one group got CP201 twice a day, 15 milligram per dose, twice a day, also for 12 weeks. And what you see here is that the high-dose group actually remembered better pictures. So they were shown one picture, and after 12 seconds, they were shown 4 pictures, and they had to choose which of the pictures they saw. And it's like, like an intelligence test. And those that were treated, as you see here, remembered better even after 4 weeks of treatment. And they also remembered better if after 4 weeks of cessation of treatment, which means that there was a dose-dependent, long-lasting effect. So this trial actually prompted additional trials, which I will tell you about another one in a minute. So I'm going back into the mechanism for one point. And so I told you that the mechanism is associated with the microtubules and also with this cleaning system that is called the autophagy system. And this autophagy system has been directly linked to neurodegenerative diseases like Alzheimer's disease and Parkinson's disease that we heard about this morning. And I'll go fast into this one. So this is the cleaning system, and what we showed is that ADNP binds to this cleaning system, that it makes like vacuoles that clean the precipitating protein, and NAP and CP201 enhances this binding to this protein that ADNP interacts with directly. So NAP actually enhances the binding to the microtubule and binding protein and also to the autophagosome, and you need the 2 systems to interact. And the previous slide was my science to show that actually we showed it in the test tube and in cells. And what we found, our claim to fame in the schizophrenia, so now I'm talking about totally another disease. So schizophrenia, it's not a disease of the aged, but it is also a brain disease, even though it's a psychiatric disease, but you know that both autism and schizophrenia can actually be affected by something that happens during pregnancy. Autism is something that happened during early pregnancy. Schizophrenia is late pregnancy. So schizophrenia could come because of some viral infection during late pregnancy. In any event, this cleaning system, the autophagy was all the time associated with neurodegenerative diseases. Schizophrenia was originally thought about something that is psychological. We don't know anything about, and it's psychological, it's in the air. But apparently it has a very important organic component to it, a molecular component to it. And what we discovered is that not only neurodegenerative diseases, but also in schizophrenia, the autophagy system is misbehaving, it is deteriorating. And we looked at a protein that it is called Beclin-1, which is the initiator of autophagy, and what we saw is that it is decreased in the brains, mainly in the hippocampi of schizophrenic patients. So this was the major finding, and ADNP has a brother, which we called ADNP2, and the expression of these two also changes in the schizophrenia brain. So we found here another connection, and we found that it's not, and I have to add, so Beclin-1 is dysregulated in Alzheimer's disease, in Parkinson's disease, now we show in schizophrenia, and also this system is dysregulated in autism. So there are common pathways that go in these diseases: development of the brain and deterioration of the brain, and then death of the brain, like in Alzheimer's disease. And we find also in the serum, we also find a change in, or more in the blood cells, we find a change in ADNP in these schizophrenia patients, as you see here. So we find more, actually, as if the system is trying to correct itself. And in an animal model, we found, in an animal model of schizophrenia, when we look at the brain of this animal model, and this animal model of schizophrenia actually has a problem in microtubules. There is a protein that is called STOP, which is associated with, or MAP6, microtubule associated protein 6, which stabilizes cold stable microtubule. And this protein is going down in the schizophrenia patients. And what we found in this model that shows schizophrenic behavior, that Beclin, this protein that regulates autophagy, so I hope I'm not mixing you up, is going down. And when we treat with NAP, we actually increase it to the normal situation. And when we treat with clozapine, we don't. So clozapine is a drug that is used in schizophrenia, but it doesn't affect this reduction in autophagy. On the other hand, it actually can enhance autophagy, which is sometimes, what we see is actually a reduction in autophagy. And what I'm saying is that NAP enhances autophagy, and clozapine can reduce autophagy even further on. So, and this is associated now, the protection of NAP is also associated with protection of the cognition in the mice, which you see here. And I'll go fast just to show you that there was a clinical trial showing that given to schizophrenic patient, our compound, which is also now, there is another name, it's the same compound, davunitide, increases functional activity of daily living. And you see here, and here it was more even apparent in the low dose. So one dose daily for 6 weeks or 12 weeks increases activity of daily living, like the ability to write a check, the ability to ride the public transportation, and so forth. So where the drug is going now is to the ADNP syndrome children, and the clinical trial is now being planned, and hopefully it will start soon. I got a prize for that. I was the champion of hope in the Science International in California last year, and this is the management team of Coronis, which is together with me trying to develop. And I also, Karen gave me also a prize for best scientific papers, and I love this. And I'm trying to do it every year. These are my papers. This is my lab, 63 students over the years here in Tel Aviv University, and I love Tau. Thank you. So I don't know, no questions?
I know I'm on the scientist side, but I think you brought the two sides very nice together. So it's open for questions. Please.
Just a clarification. You said tau precipitates, but tau is never in solution, is it? If it's in a tubule, then it comes out of the tubule and just goes
So
down.
what happens, tau on its own actually doesn't have a structure. So yes, it binds to, it is usually not soluble, although it can be soluble in the cytoplasm. So it either on the microtubules, but what happens is when it's hyperphosphorylated, then it falls off the microtubules, and then it forms these precipitates. What we don't know exactly is how this happens. So we don't know what drives it. Is it because the microtubules are shorter and it falls off? Why is it all of a sudden falling off the microtubules and forming these neurofibrillary tangles? But then it has actually a beta sheet structure, so it changes the structure.
Forgive me for harping on this, let me just try this again. To precipitate means something is in solution, dissolved, and then comes out of solution.
Yeah.
Is that what happens to tau, or does tau just come off the tubule and then just fall through the solution? I'm asking because I can think of potential different therapies depending on whether the tau is transiently in solution or never dissolved in solution and just falls out.
In the test tube, it is in solution. So I'm not so sure, like in the cytoplasm, I think it can be in solution, but what people are trying to do, and I'm not sure this is the right way to go, but what people are trying to do is to dissipate these aggregates of tau. And what I asked, if you listened in the morning, what is now very much kind of in the forefront is that it precipitates, it forms aggregates, and then these aggregates can move from one cell to the other and have some kind of a prion effect. And that's kind of the common theory now. Okay. All right. Thank you.
Yeah, I'm going to ask a quick question, but it's a little more broad if we're talking about some of the context of all the things that we're talking about. So this is one molecule, right? This is one gene. We have about 20,000 genes. Well, depending on if you're looking at RefSec or where you're looking, we have about 20,000 genes, 1 million isoform proteins. But this seems to have a huge impact, right? I mean, it's first of all, when we talk about autism, which is we know is pervasive and is becoming a bigger problem worldwide, so maybe it's not on the healthy aging spectrum, but it's still a major concern to all of us. And then, of course, on the aging perspective. So if you would look at the genome, how many genes do you think have such an impact? And when we're starting to think of therapies, are we starting to think of a cocktail of a number? Are we going to have to individually find each one? What do you think of kind of a big picture approach when we think about healthy aging?
So this is very important because, like for example, if I take the autism world, so there are like 800 genes or something, depending who you talk to again, that can undergo mutation, and then there is autism, but maybe there is one case or two cases, but then there are like 10 genes that are up there. And and then if you take like each one of them, they will have a very big impact. So yes, you can kind of categorize and have some genes that are much more impactful than others, and but when you tailor for aging, you probably need to look at the broader picture. So one gene won't be enough. You will have to have more of a mixture, because like the idea of vaccinating against amyloid, it didn't work, up to now. And it cost billions and billions of dollars, and it didn't work. And people still are trying to vaccinate against the amyloid, even though it doesn't work. But maybe a combination with understanding the basic mechanism, with thinking about that we lose the microtubules all the time. Maybe we should protect also against that, but maybe just protecting against that and forgetting about the amyloid is not enough. So there you are perfectly right. You need to look at the whole picture. Yeah, thank you.